Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Epen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@epen.info. My guest today is Professor Brian Wandell, who is a professor at the psychology department of Stanford University. He's also a member of electrical engineering, ophthalmology, and graduate school of education. He's the founding director of Stanford Center for Cognitive and Neurobiological Imaging, and he founded the Stanford Center for Image Systems Engineering program. Professor Wandell's research centers on imaging science and technology, spanning neuroscience measurements of the visual cortex and reading development to simulation and design of imaging systems. Welcome, Brian.
Thanks, and thanks for the invitation, Gil.
I was a bit like a kid in a candy store, Brian, looking at all your papers. I guess you're working the graduate students pretty hard at Stanford these days.
Stanford's in a confusing moment with the campus being completely shut down, and we're all kind of wanderers out here.
Yeah. I want to start with one of your recent papers. It's entitled The Human Connectome Project for Disordered Emotional States, in which you say, through the Human Connectome Project, HCP, our understanding of the functional connectome of the healthy brain has been dramatically accelerated. Given the pressing public health need, we must increase our understanding of how connectome dysfunctions give rise to disordered mental states. Could you talk a bit about that paper?
Yeah, I'd be glad to. Thanks. Actually, that whole project, although I participate in it actively, I want to give a shout out to my colleague, Lee Williams. She's a terrific psychiatrist.
Yeah.
And has the reason why I'm involved is because I have certain skills in being able to use magnetic resonance imaging for measuring and quantifying individual human brains.
Yeah.
And I've always been admiring people like Lee who are working on this just huge problem of people being emotionally stressed. I think these days all of us can relate to the kinds of stressors that are really roiling our country. But even in the best of times, the greatest global disease burden in Western countries, I shouldn't have said global, but the greatest disease burden in Western countries for people from about 20 till the ages of 60 or so are really anxiety, depression, mood disorders, and so forth. And it's not until later that cancer and heart disease start to dominate.
Right.
And so the goal of Lee's project and the way I try to help is, you want to be able to look at a single person and measurements for that person of what's going well and what's not going well, either with their brain structure or function.
It's sort of looking at the, I was surprised, Brian, to look at the numbers here. So it says mental disorders arising from high levels of negative emotion or from the loss of positive emotional experience affect over 400 million people globally. So the idea here is to look at sort of the wiring of the brain and then have some hypotheses.
That's right. And also, it's not likely to be the case at something like this that everybody who has problems are the same.
Yeah.
So the idea that you take a bunch of people and average them and try to see what's happening is, has proven not to be a good approach. So maybe you or anyone who's listening might have heard of this notion of kind of precision healthcare.
Yeah.
And that's the idea that you would learn about this particular person. Sometimes it's through genomics, but sometimes it can be through brain imaging.
Right.
And see, is there something about this particular person? We'd like to be able to look at a person and say, one of the things Lee uses, there are a few interventions that can help, some behavioral and cognitive and some pharmaceutical. And the amount of time that it can take to get to a helpful solution for somebody can be quite long, and it can be quite an unpleasant journey. Mm. And so shortening that time in the cases we can help is one important aspect of what Lee and our team is trying to achieve.
Yeah. So this is through MRI scanning, right? So the brain has a template, but like you say, each individual is different. Yes. But you are sort of looking for patterns in those scans that might tell us something. That's
exactly right. The other thing you should know, and not everybody would know this about magnetic resonance.
Yeah.
It is one instrument. In other words, there's a big magnet, and there's a bunch of electronics, and there's a bunch of software that we use to analyze the data. But it can be used in an extremely general way. It doesn't only measure one thing. And you can control the instrument to measure, for example, the white matter pathways. These are the long axons that connect different parts of your brain.
Yeah.
Or you can use it to measure the gray matter of your brain, which is where the neurons, the cell bodies are located. How thick is it? What's their density? Or you can use it to measure indicators of functional activity, what is responding, which neurons are responding during that task. So it can be used in many, many different ways. And that's why this project has so many authors, and it's a really big challenge. We're trying to use MR in a variety of different ways to see which signals might be the most helpful for shortening the diagnostic time.
Are we generally successful?
I would say there has not been. I'm very optimistic.
Yeah.
About the next 10 or 20 years. I'm old enough so that I'm not going to, be doing the last 20 years of the next 20 years of this. But the physics of this is getting much better. The ability to handle large data sets at scale is getting much better. I look at the younger scientists, and they're just a lot smarter than I was. And I am very hopeful that we will come to new ideas. But I would say at this point, there have not been real breakthroughs in diagnosing mood disorders via standardizable MR techniques or other techniques, either EEG. But I would say the number of companies that have started up in this space and the number of hopeful little papers, gives you a sense that we might be making some progress. But I can't really say that I'm confident in any of them at this point.
Yeah. No, it's interesting. I mean, on one hand, you can really understand how a single cell could become a human being. On the other hand, you say, well, there is a template, a design template. How many variations could you have? And I guess the answer is infinite. And hence, it's a really, really complex problem to solve. But if you can solve it, in the healthcare context, Brian, as you know, we treat mental health almost separately from physical health. And there is, really good data that shows that there is a huge connection between mental health and physical health. So any diagnostic modalities that could surface these things earlier and earlier intervention in the mental health arena could have a huge economic impact on the healthcare system.
That's 100% true. I really agree with that in all parts of what you said, that even the mechanisms from what we consider physical health, meaning things like a good vascular system, good posture, good getting out and walking, and mental health are quite significant. Those are some of the things we know best. By the way, these aren't magical effects.
Right.
So the thing, when you look at a brain, so in neurosurgery, the brain is just filled with blood and vasculature. And when you walk and you keep your heart pumping and your vasculature is in good health, that helps your brain health. And we don't have the details of how that works, but there's no doubt that a good blood supply, oxygenated, your heart pumping, just matters enormously for brain health.
Mm-hmm. Yeah, it's a fascinating area. I want to jump into another one, which is related, Brian. So this is entitled Biological Development of Reading Circuits, Current Opinions in Neurobiology, where you mentioned brain computations operate over a range of temporal and spatial scales. Understanding action potentials and synaptic efficacy is essential for understanding certain aspects of performance. You want to talk a little bit about that?
Yeah, thanks. So let me give a shout out. That particular collection of papers, I think that's a review. There's a student of mine, Jason Yateman, who went off to Seattle for a while as the professor there, and he's now back at the Graduate School of Education at Stanford. And he and Aviv Mezer, who's at the Hebrew University in Jerusalem, and I were really curious about how come some kids learn to read and some kids don't. And I have to say that the first thing we learned as we started looking at that field was, for the vast majority of kids who have problems learning to read in the US, you could take care of a lot of those problems if they just got a good breakfast and felt safe in their neighborhoods. And it was just, we just improved the general conditions of their lives.
Yeah.
But beyond that, and these are the ones we study, there are kids who just, they're motivated, they're smart in many other ways, and they just can't pick up the tricks that it takes, particularly to learn to read English, which is a very tough language in its own way. And so we started looking at that using magnetic resonance imaging with the same idea. Could we do something helpful so that we could look at one kid, same principles we spoke a moment ago.
Yeah.
Could you look at one kid and say, oh, this kid, he's gonna be okay in 6 months. So you tell the parents, just keep working, keep her doing her homework, and it'll snap in a few months from now. But this other kid say, yeah, we've seen this pattern, and it could be like some of these very famous and yet successful cases. Maybe the most famous these days is Charles Schwab, who's very successful. And we would like to be able to tell the parent, well, in this case, you're really going to have to get them educated in a different way. Mm-hmm. So that particular point that you spoke to, when we got into it, we found a couple of just forces in the neuroscience field that we felt we needed to address. And one of them was that neuroscience as a field has a massive focus on single cells and the synapse.
Yeah.
And, when you measure MR, you don't measure single cells. The spatial resolution for magnetic resonance is, you're pretty good if you pick up a cubic millimeter of the brain,
Oh, wow. Okay.
which is going to have about 50,000 cells.
Yeah.
And amongst all these, high-end electrophysiologists and basic scientists, being at the synapse, and there could be, 5,000 synapses on a single cell. That's where the cool people are. Well, that's, if you're a computational person, as I am, no single synapse is really critical for computation. It can be critical for drugs.
Yeah.
And a lot of this comes from big pharma. So, we got to control the synapse because that's where we can deliver drugs to and have an impact. But for understanding what the brain is doing, sometimes you want to be at a different spatial resolution.
Right.
You want to see different parts of the brain, the back to the front of the brain. You want to see the pathways that connect it. And so that paper was one of our attempts to express the view that there's real meaningful work to be done at the coarser spatial resolutions.
Yeah, that's really interesting, Brian. So, you mentioned sort of the initial conditions that the kid is in, and it has a huge impact, right? You say he has cell development, growth, and pruning of dendritic arbors and axons, the status of the glia. So really at that scale, not at the cellular level, but at the structural level, if you don't have the right initial conditions, you are kind of designing in a sort of a different system, almost sounds to me, right?
That's right. Yeah, and in fact, you raised let me add a final word for everybody. Most people know the word neuron as the kind of computational element in the brain. But over the last, I don't know, maybe not more than 20 years, led by a quite remarkable scientist who was here at Stanford, Ben Barres.
Yeah.
And more recently, somebody who I don't know well, I've met her, but whose work is just fantastic, Michelle Manji at Stanford, and others around the globe. The importance of a completely different cell type, of which there are almost as many in the brain, these glial cells, has risen in terms of our understanding of how important they are for the development of the brain, for healthy signaling in the brain, and again, the ability to distinguish the health of the neurons from the glial cells, from as we were saying before, the vasculature, whether the blood supply is getting there.
Right.
If you care about a kid's health and whether a kid is going to develop and learn to read, you want to be able to assess all of those systems, as well as the neurotransmitters and the molecules. So, a little bit of all of us not standing on one another's toes, but all of us sort of helping one another see the different parts is something I think it could happen. I think is maybe starting to happen. And I hope we all participate in working together at that and seeing the global picture, not just the picture from inside the synapse.
Yeah. And even at the MRI scale, so cognition, and you mentioned this before, cognition and white matter tissue properties is one example of how these factors may set important limits on both cognition and affect.
Yes.
So, yeah, go ahead.
Oh, I was just going to say, if you don't have the right connections between different parts of the brain, and that's the job of the white matter. If you don't have the right connections across large parts of the brain, things won't go well. So yes, that's really important limits.
Yeah, and what's your sense? I don't know much about this, but once the brain is set in that context, can you really intervene and make it better?
Yeah, so that's a very important question. And sometimes you'll hear the phrase brain plasticity.
Yeah.
In that regard, and one of the big topics in neuroscience, in addition to developmental plasticity, is the question of brain plasticity in the adult.
Right.
And it was a very heated area.
Mm.
So in the early days, so there's a kind of a pair of very famous Nobel laureates, Hubel and Wiesel, who did many studies of brain development and came to the conclusion that there was an early stage of plasticity, which we all marvel at, right? We watch our kids grow and we understand that's hugely important. And kids are remarkable what they can recover from and can't. And then they argue that there was a critical period after which there wasn't much plasticity left.
Mm-hmm.
And there was then a big pushback against this, that no, the brain is plastic. And there's a period of time, I don't know, Gil, if you remember this, but the public radio stations used to have ad after ad after ad about brain plasticity and companies that would help you drive forever and live forever.
Right, yeah.
But the extent of plasticity in the adult brain is far less than the amount of plasticity in the developing brain.
Mm-hmm.
And that seems to me pretty clear. There are still people who would argue with me about it, but I think that the limits are pretty obvious. And so the question now is, well, when there is damage, is there something you could do to reintroduce plasticity?
Right.
Either in the form of a behavioral intervention that would release whatever is blocking your brain from regrowing, or in the form of it could be pharmacological or electrical stimulation. And this matters for cases like stroke patients who've had parts of their brain destroyed. It's open to technologies like stem cell therapies, where you would try to reproduce and regrow and reinsert things. There's other kinds of ways to grow bits of brain called human brain organoids that people are trying to achieve. So in any event, there's a lot of lost plasticity, and there's a lot of interest in being able to literally repair the human brain. And it's wonderful topics and very, very, very early stages on this.
Yeah, and this is not the paper, but I know that you have done some work in this area too. So certain diseases like Alzheimer's, for example, one question would be, is it really destroying any remaining plasticity in the brain as well, right? What is the latest thinking around that?
The Alzheimer's work, so that's called the field. It's within the broader field of neurodegeneration.
Yeah,
and that's a kind of brain plasticity, but it's the kind that you don't like.
Wow.
So the brain is changing, but it's changing in a way that's not helpful. And so reversing that change, putting on brain plasticity, but undoing the effects of the destruction from Alzheimer's, in which this synapses are lost and communication is lost and so forth, is important. And you're raising, so people have now done many fundamental, important experiments in animal models
Mm-hmm.
to reverse the effects of the plaques and tangles and the degeneration, and to try to bring back actually performance levels in these animal models. But so now you've taken us into an area where when you succeed in reversing something that seems, and model is important. It's a kind of a model system. It's not the same as the human brain.
Yeah.
The mouse brain is 1/3000th the size of the human brain.
Right.
And so it's going to have all kinds of differences. And it's kind of one of the tragedies of our, current standing in the neuroscience of this, that none of the interventions that have been successful in the mouse have effectively translated into human.
Yeah.
And that's why one of the reasons why I'm so passionate personally about trying to just do better with the technologies like MR, that we can both measure and potentially even intervene in the human directly so that we might have a better model, not have the mouse model of human, but maybe have some cases in which we have human models of human. And anyway, that's been one of the hugely important and also frustrating aspects of what we look at. So at Stanford, we have we have a very broad, not just human, but a very broad neurosciences institute.
Yeah.
And Clara Wu and Joe Tsai donated, and it's called the Wu Tsai Neurosciences Institute. And the ability, mood disorders are important, as I mentioned, Lee's work and a number of others, but the Alzheimer's part of that is also very important. These are huge financial burdens on society, and more than that, it's not just the money. It's seeing some.
Yeah. I mean, the space you're in, Brian, at the intersection of computer science and neuroscience, I sometimes feel like as we make progress on the computer science end, the brain becomes more and more enigmatic, right? It's almost like God has a sense of humor. You get teased that you found something, but then you find that there's 1,000 other things you need to figure out.
That's interesting. Could you say a little bit more about that? I kind of get what your point, but say a little more about that.
Yeah. So what I was saying, Brian, is the computer science, we are making quite a bit of sort of, I would call it mechanistic programmatic advances. So the latest thing in terms of deep neural networks, we know that the brain doesn't learn that way, but we are making great progress in neural networks and so on, right?
Yes.
But when you go back and look in the brain, it seems like we find more and more things that we don't understand.
Right.
Right. We at no point we can say we have a better handle of this, and we know this is how it's functioning. It seems like we have some component level understanding, but it's not really progressing to more of a holistic understanding.
Yeah, no, that's important.
And I wonder, so we take this reductionist approach, right, in science. And I often wondered if that is probably the wrong approach to the brain. And you kind of hinted on that as well, that it's the structural aspects of the brain that is really driving it. It's not necessarily the cells, the cellular type understanding.
Yeah, I hate to. So I am quite fond of and count amongst many of my close friends people who really do important work on studying cells and their behavior. And I wouldn't, I'm sure you also, neither of us want to diminish that. I just felt that the balance had gotten to the point where people would say, if you were studying the system, you weren't a real scientist. You were just, joking around. You needed to know the genes and the molecule.
Right.
And I thought that had shifted a little far. And I would never argue that you don't want to know the genes, or if you could, that of course you would want to know that. But, rebalancing a little bit so that we keep the whole human as part of the measurement system. And, it's miraculous what you can do with genetic manipulations, these CRISPR technologies, optogenetics, and these are miraculous things.
Yeah.
But they're not quite this, and they're a great complement if we keep the patient or the developing child who's learning to read. We need to keep them in the lab. We can't just say, oh, it's all going to be about these cells that we get out of a mouse.
Right. Yeah, I want to jump to another area, Brian. So you are doing a lot of work in imaging systems, camera designs related to artificial intelligence. In one of the papers, you talk about a digital camera and fluorescent signals as sort of the first application in oral cancer screening. Could you talk a bit about that?
Sure. So, for me, MR is one of the imaging modalities, but as you say, I've been fortunate over a kind of long career to be able to work on many forms of optics and digital image sensors and so forth. And when I see a medical application, it kind of brings it all together for me. And fluorescence is one of the kind of fundamental properties of certain kinds of healthy cells. Mm-hmm. And so measuring fluorescence can be hard. And so that for me, right, that's my job, try to find something that's a little hard to do.
Yeah.
And make it easy.
Yeah.
And so one day I actually, so this is a project that was actually kicked off by my son, who's an oral surgeon in Texas.
Yeah.
And he was seeing my wife and I were looking at a particular device. He said, if you could take a picture of somebody's tongue, mm-hmm. and I had never thought about tongues. You could take a picture of somebody's tongue and see the fluorescence coming out of it, and I'll define fluorescence in a moment.
Yeah.
Then you could really get an early diagnosis of cancer in the tongue.
Right.
And I had never heard of anything like that. And he said, but and that's not that big a deal here in the US, but it's, matters.
Sure.
And then we came to learn that tongue cancer in China and India is a massive problem because of smoking and perhaps other factors. It's really...
Yeah, smoking plus the alcohol, I think, has a huge negative effect, yeah.
So designing a camera, a lot of my world is about software and software simulation and designing cameras and devices using software. And so we started to think about how you would design the lighting system, the filters that would receive the light, the sensor, the processing for a camera that you could just poke into somebody's mouth briefly and take a picture and get a sense of where the cells were fluorescing or not. And fluorescing means that you put in one sense of fluorescing. So it means you put in light, let's say blue light, but when it comes back, it's red. And normally when you put, shine blue light on something, what's reflected back to you is blue light.
Right.
But in many cases, the fluorescent cases, you put in the blue light and there's a chemical process that arises, a physical process that arises in the cells that changes the wavelength of the light, and it will return the lower energy state. And so the short wavelengths will end up being longer wavelengths, which are lower energy. So how you would design the filters and the sensors and the cameras and the processing for that's the kind of thing we do for fun. And building a camera like that was something we were interested in. And I had another former student I want to shout out to, Feng Zhao, who's started a company called Feng Yun Vision in Beijing.
Yeah.
And Feng said, well, he would be thrilled to go and just build us test equipment. Because if we could make some progress on this, he felt it would really help with oral cancer in China. And so that's what we've been doing. He's been building little prototypes for us. We have kids in California and a kid in London and some people in China. And we're all passing around software and building the device, taking test measurements and starting to write papers. And we're learning about the literature. It's kind of late in life, but I'm reading fantastic work from people out of Houston and Dallas who've done some early experiments on this. And we have a prototype camera up now. We're taking pictures and we're trying to see if we can make it more and more robust. And then if it works out, we're going to put it back in the hands of a colleague here who's an oral surgeon, and also in the hands of our son who's in Texas, and see if we can't move it into the clinic. And we're not very far along, but we have some early preliminary measurements that look promising.
Yeah, this is very interesting, Brian. So so you're not really talking about any sort of deep learning needs here. You actually can write down some heuristics, basically saying if I get this type of fluorescence, then I have this probability that tissue is healthy or otherwise, right? You don't really need to use any AI techniques even.
It's what you're raising is actually very interesting and important to me. Certain cases, you just have the algorithm. And I would not say that we're sure yet
Okay.
that how we will be able to do this. So, I would be happy to talk to somebody like you, for example. I don't know whether the spatial pattern of the fluorescence that comes off of the tongue, if it's all on one side versus kind of little dots spread across the tongue,
Right.
does that matter for health and diagnosis? Well, I don't really know.
Yeah.
And it could be, when you and I spoke earlier, we spoke to one another about collecting and organizing enough data
Yep.
so we can look at these things. And we're keen to work with people like you and others who are skilled in automating the search for signals through machine learning.
Right.
To answer that question.
Yeah, and that is one of the other things that you're pursuing, Brian. So you have a company, flywheel.io, and one of the issues in machine learning, multiple issues, and one of them is obviously related to data. How do we collect and organize data in a systematic way so that experiments run on that data could be reproduced by anybody who is using that data. And that's a very important aspect, because as machine learning and deep learning really kick into mainstream, it's really easy, I would say, to build models given a bunch of data. It's not that easy to actually reproduce those results in a robust fashion, right? That's what you're really focused on in Flywheel.
Yeah. Thanks. Yes. So let me say it came about because when I started, I guess it's been about 10 years when we were fortunate to get our first magnet and start the MR Center. And we knew we needed to take care of the data. Because when you first write the check for one of these things, you realize, wow, that's an expensive instrument. I better not mess up with managing the data that comes off of it. We have about 40 different labs at Stanford who use the instrument.
Mm-hmm.
And so we worried a lot about stewarding the data, making sure that we didn't lose it, that it was properly recorded, that it would be easy for people to share if they wanted to share. And these weren't going to be my data. They were going to be data that, different labs would acquire. And, they weren't always ready to give it away right away. They wanted time to process it and work on it. But we knew there'd come a point where they'd be done and it should be easy for them to share and to compute, try things with colleagues in electrical engineering, computer science, applied math, with these data. So yeah, we built you would have thought, by the way, I don't mean to throw Stanford under the bus here, but you would have thought at a place like Stanford, when you buy a big expensive instrument, that it would be an instinct for them to say, whenever we put up one of these centers, we always have to have a data management plan.
Mm.
And Stanford was actually really great about helping us choose the color of the paint and the carpets. But man, when it came to data management, it was one big shrug.
Yeah.
So my buddies who were building this thing with me, particularly Bob Doherty, who's now at another company called Mindstrong, and Gunnar Schaefer, who joined me at Flywheel, we thought we really had to build a data management system. We actually tried to get help from our colleagues in the computer science department. That was another interesting experience, because I went over there, and I'd been around long enough, and they would take meetings with me, but they'd all look at me and say, well, that's just too easy. That's not a hard problem in computer science. In computer science, we are doing these super advanced things.
Yeah.
So we fell in between the, just could buy it off the shelf from Dell or something, and something that computer science people would work on. So we built our own system for data and computational management, and it seems to have fallen into a sweet spot. And I'm glad you see the value in it. Many people do. At this point, Flywheel's doing okay. But for us, the main point is being able to share the work and letting other people check your work. So, a lot of stuff is hard, and we all benefit if when you publish something, somebody else can come in and check that you did it the right way,
Yes.
and then build on it. And that really is the premise of that company.
Yeah, yeah. The other side of data, as you know, Brian, is, when I talk to my clients, sometimes they say, well, we have to collect all the data first, and then we'll figure out, how to use it. And uh, oftentimes, you spend years collecting the data, and by the time you come to use it, the data has already changed.
Yeah.
And so the real, capabilities in the area of machine learning and deep learning is really the ability to systematically discard data. And this is not something people really think about. I think the brain is a good example of it. Brain is a very efficient discarder of data, and focus on what is really needed. But our kind of designs in the AI arena basically is looking for all the data in some way, right? But the data is no, if the data is organized in a form that is standardized across different modalities, then you will be able to see what is most useful much faster, right?
I agree with you. Yeah, I have to say that I think the reason there are two things that we've learned so far, and I hope the story will continue to. We have a ways to go, but there are two pretty fundamental things we learned that build on your comments here.
Yeah.
The first is don't let the data out of your hands when you get it from the instrument.
Yes. Yeah.
As you were saying how hard it is to go get the data. Well,
Yeah.
if you're sitting at the instrument and the first thing that the instrument does is put the data in a database,
Right.
you're okay. And for many years, and still many people don't appreciate this, if you say you take the data and you let it go out to the 40 different labs, and then you say, well, could I have it back, please? That's impossible. So by putting the acquisition of the data right in the instrument itself, which we could do on our scanners, that was very helpful. We now have this, just in our one center, we have a massive data set, but we're now managing. I thought it was massive, but I would say, I don't know if I'm allowed to, I may not be allowed to name the company, but we're working with a very large pharmaceutical company.
Yeah.
And they have millions of data sets. I was impressed that we had many tens of thousands. But these companies at global scale have millions of data sets, and they're not organizing them well.
Oh, yeah.
So getting the data in and organized is, key. And we're glad to have this one partner who recognizes that, and hopefully go others. The other thing that we learned is that in each field, so we're particularly comfortable because of my background in medical imaging. But I don't really think that exactly the same format and how we store it and the metadata about it and so forth will work exactly the same way in all fields, even other types of medical imaging. So I'll say, I do a lot of MR, PET, EEG, things of this sort, ultrasound. But when you go to digital pathology, it's a slightly different culture.
Right.
So the other thing we've learned is that when you build these things, you have to be aware of the culture of the enterprise. If it's pharma versus medical instrument makers who make wearable watches,
right?
Just different organizations, and we need to keep learning what works properly in the domain-specific organization.
Yeah.
Anyway,
yeah.
It's been a very, thanks for letting me talk about that a bit. It's been very, I do think you're right. You've said this a number of times, that the acquisition and organization of the data is really important, and that's often undervalued by scientists and engineers. So anyway, thanks for
it's often undervalued. And I think you talk about this in one of the papers, Brian, which is, if you are embarking on a process, take a lot of time to think through the data that you want, right?
Yeah.
Because otherwise you'll be cycling through this process over and over again, trying to make slight refinements to the data capture process.
Yes.
And that is really costly. I saw one other thing that I thought was also very interesting, Brian. So this neural network generalization, the impact of camera parameters, another paper that you have, where you say images coming out of different cameras from a neural network. This is convolutional neural network. From a generalization perspective, is roughly the same as the generalization you get from images in ray traced multispectral synthetic images. What exactly are those?
Yeah. It's very hard to build a camera. It's expensive.
Yeah.
Camera and run it out and test it. And so, just like it's hard to build an MR scanner and test it. So in imaging, there's a little bit of a movement that some of us are, trying to get going, to use simulations, but physically accurate simulations of the devices, so that we can build, say, complex driving scenes and then theoretical cameras in simulation and drive them for a couple of million miles without having to actually build the novel camera and put it out on the road and drive it for a million miles. Elon Musk can do that, but
Yeah.
and so that particular paper was asking, how close are we getting?
Right.
Are our simulations, which the technology for getting physically accurate images that you can use as inputs to physically accurate cameras, it would be called multispectral, because it's many different wavelengths of light. Ray tracing is the right technology for getting physically accurate measurements.
Yeah.
So can you do multispectral ray tracing through optics onto sensors and train neural networks based on that, so that you can do it all inside of Google Cloud or on Azure or Amazon or something, and get a pretty decent assessment of how well it'll work before you put it on somebody's car.
Right. And at least in the first few attempts, they do pretty well.
There's a little growing in, So I would say right now, so the thing, we didn't know how to measure it exactly, right? Because we weren't I'm kind of practically oriented, and I couldn't say, well, okay, this is how it's simulated. Now let's get out there and drive for a couple of years.
Right.
And that was not available to me. So what we could do was say, well, people had kindly shared large data sets. That community is pretty great about sharing data sets, the machine learning community.
Yeah.
And so we could say, well, suppose we trained on the data set from Berkeley, and then we tested on the data set from Germany, or we trained on the one from Germany and tested on the one from China. How well would they generalize?
Yeah.
And what we found was that the generalization between data sets people had posted and were acquired was no better than the generalization between the synthetic
okay.
that we would build. In fact, the synthetic data set, could predict some of the real ones better. It was right in the mix. So that gave us encouragement that we should keep going. So
yeah. That's exciting. So in conclusion, Brian, if you look forward 5 years, where do you think we will make the most advancement? If you think about cognitive and neurobiological imaging, some of the things that we talked about today, where do you think, what is the area that you're most excited about? Let me ask it that way.
I'm on this mission. So let me say my partner in this is in the neurosciences is the director of Clara Wu and Zhou Tsai's Neurosciences Institute at Stanford. The director is a guy named Bill Newsom.
Yeah.
And we were fortunate enough to be able to put up a building to house the Neurosciences Institute. And I'm going to tell you what we bet on.
Yeah.
And this is a dangerous bet. But people were really annoyed with us, but we stuck it out. In the design of the building, we implemented 2 floors. We kind of literally merged two floors, cut them out, put them in the hardware of places where theorists would sit and think. And normally, when you build a building at Stanford for neuroscience or almost any biology, it's one lab bench to the next. It's hoods for maintaining chemicals, and places for animals and so forth. But Bill and I, with backing of others, got space built into the building that's going to be really hard to reconfigure for anything other than theoretical neuroscience.
Right.
And our feeling is that the number of experiments is infinite, and if we want to make more rapid progress, we're going to need people like you or other theorists who sit and think about the problem and develop theories and develop models to guide both the massive new data sets that are being acquired and what we should acquire next. So that's the bet we made. I don't know if it's a 5-year bet or a 50-year bet, but the bet we made is, and we done our best to try to protect the space so that there will always be a place to think about how we should organize the data rather than massively go and collect a lot of data. Some people are annoyed with us for it. Some people applaud us for it. I hope it works out.
Yeah, that goes back to your comment about culture, right? So at the end of the day, progress is really very highly correlated with culture.
I really have to say thanks for saying that, because you remind me, one of the things that made us go for this was when we did this institute. Bill and I and a couple of others had, I don't know, about 20 different dinners with people from all around campus, groups of 10, 20 engineers or people from the ed school, or just trying to figure out what we thought was important, what they thought was important. And one very memorable time was when somebody started yelling at us, basically in a polite way, but yelling, you need to do mathematics for this. You guys are never going to get anywhere until you start understanding the difference between theory and model, and you need mathematics and stuff like that. And we were prepared for that, and I think we acted on it.
Yeah, that's excellent. Yeah, this has been great, Brian. Thanks so much for your time today, and good luck with everything that you're doing at Stanford.
It's been a pleasure meeting you. Thanks for the opportunity to have the conversation.
Thank you. Bye.